There is a moon in our solar system that is currently erupting. Right now, at this moment, it is blasting a column of warm ocean water 800 kilometers into space. That water contains salt. It contains organic molecules. It contains hydrogen. It contains phosphorus. And it falls back down as snow. What is in that snow? That is the question that has rewritten the entire field of astrobiology. That is the question whose answer, when it came in 3 separate waves of discovery across the last 3 years, stopped physicists mid-sentence, sent the paper to the top of the download charts at Nature, and caused at least one senior NASA scientist to describe it as the most consequential suite of findings since the discovery of water on Mars. We are not talking about a faint chemical trace. We are not talking about wishful modelling. We are talking about the complete molecular checklist for life as we know it. Found, ticked, and confirmed inside the active plume of a moon that should, by every prior assumption, be completely dead. The moon is called Enceladus. It is smaller than the United Kingdom. It orbits Saturn at a distance of 237,948 from the planet's centre. And it may be, this is not hyperbole, this is the scientific consensus of 2025, the most likely place in the known universe, beyond Earth, to harbour living organisms. To understand why Enceladus is extraordinary, you first need to understand why it has no right to exist the way it does. The moons of the outer solar system are, as a rule, frozen. Not metaphorically frozen, not the way we say a city freezes in winter, meaning it gets cold and inconvenient. Frozen in the way that means geologically silent, chemically inert, and billions of years removed from any meaningful internal process. Out beyond the asteroid belt, at distances where sunlight is reduced to a faint cold glimmer, roughly 100 times dimmer than what we receive on Earth, there is simply not enough solar energy to warm anything meaningfully. The outer moons were expected to be dead rocks coated in ice. Most of them are, Enceladus is one, 272 billion kilometers from the Sun, a number that should seal its fate. At that distance, the surface of the moon sits at an average temperature of minus 198 degrees Celsius. In practical terms, that is cold enough to freeze nitrogen. It is cold enough to render most chemical processes essentially motionless. When astronomers first catalogued Enceladus in the 19th century and measured its extraordinary brightness, its albedo of 0.99, meaning it reflects 99% of all incoming sunlight, which makes it the most reflective body in the entire solar system, they assumed it was simply a ball of fresh, clean ice. Pristine, dead, unremarkable. A cosmic snowball, it is 504 kilometers in diameter. For context, that is roughly the distance from London to Edinburgh. You could drive across it in less than a day. It sits in a region called the E-ring, the outermost and most diffuse of Saturn's ring system, a ring so vast and so faint that it was only confirmed by the Voyager probes in 1980. And here is the first thread that should have made scientists suspicious. The E-ring is peculiar. Unlike Saturn's inner rings, which are composed of ancient, somewhat dirty material, dust-coated, reddened by organic chemistry, slowly darkening over millennia, the E-ring is made of tiny, fresh, bright, clean ice particles, suspiciously fresh, suspiciously recent. As if something were constantly replenishing it, something was. But it would take another 2.5 decades before anyone knew what. The first hint that Enceladus was not what it appeared came not from a direct measurement of the moon itself, but from a calculation. In the 1980s, planetary scientists began modelling the tidal forces that Saturn exerts on its inner moons. Saturn is a giant, its mass is 95 times that of Earth, and its gravitational field is not uniform. As Enceladus orbits Saturn, the gravitational field stretches and squeezes the moon in an uneven rhythm, a process called tidal flexing. For a solid, inert rock, this produces a small but measurable amount of heat. The models predicted a modest warming effect. Modest, but non-trivial. What they did not predict, what no one predicted, was what the Cassini spacecraft would actually find when it arrived at Saturn in 2004 and turned its instruments toward this small, bright, forgotten moon for the first time. In 2005, Cassini's cameras resolved something that does not appear anywhere else in the solar system. Near Enceladus's south pole, there are 4 parallel fissures in the ice, linear cracks, each roughly 130 kilometers long and approximately 2 kilometers wide, spaced with a regularity that, in photographs, looks almost architectural. They have been named collectively the Tiger Stripes. Their formal designations are Baghdad Sulcus, Damascus Sulcus, Cairo Sulcus, and Alexandria Sulcus, and they are among the strangest geological formations ever observed in space. The first anomaly, they are warm, in the context of a moon whose surface temperature sits at -198 degrees Celsius. The Tiger Stripes register at -93 degrees Celsius, a difference of over 100 degrees detectable by Cassini's composite infrared spectrometer from hundreds of kilometers away. On a world that should be uniformly frozen, these 4 features are, in a relative sense, blazing the second anomaly. They are erupting from each of the Tiger Stripes, continuously and perpetually. Enormous geysers of water vapor and ice particles blast outward from the surface of Enceladus at velocities exceeding 400 meters per second. The plumes rise to heights of over 800 kilometers before some of the material falls back to the surface as snow, and the rest escapes Enceladus's gravity entirely and disperses into the E-ring. Cassini photographed these plumes in 2005 in a series of images that remain, in my view, among the most iconic in the history of space exploration. A small moon, lit at an angle by distant sunlight, its backside erupting with white feathers of ice that stretch off into the black. It is a sight that rearranges something in your understanding of where life might live. But photographs were not the real discovery. The real discovery required Cassini to fly directly through the plumes themselves. Between 2005 and 2015, Cassini executed 23 close flybys of Enceladus. Several of these passes took the spacecraft directly through the plumes at altitudes of less than 100 kilometers above the surface. During these transits, Cassini's array of mass spectrometers, its ion and neutral mass spectrometer, and its cosmic dust analyzer, sampled the material erupting from the cracks and returned a catalogue of compounds that nobody had expected to find this far from the Sun. Water. That was obvious, and it confirmed what the images suggested. But alongside the water was something that shook the scientific community with considerably more force. Sodium chloride, salt, the kind that only appears when liquid water has spent extended time dissolving rock. On Earth, ocean water is salty for exactly this reason. Water has spent billions of years leaching minerals from the seabed. The presence of salt in Enceladus's plumes could only mean one thing. Somewhere beneath that ice shell there was a liquid saltwater ocean, a global one. This was not a marginal interpretation. The salt concentration in the plume material, once analysed, suggested an ocean pH and mineral content broadly comparable to Earth's seas. Gravity measurements from Cassini's flybys confirmed that the ice shell at the South Pole was thin, perhaps no more than a few kilometres, and that a large body of liquid water existed beneath it. Later measurements refined the picture. The ocean was not localised but global, wrapping the entire rocky core of Enceladus in a layer of liquid water somewhere between 26 and 31 kilometres deep. For comparison, Earth's deepest ocean, the Challenger Deep in the Mariana Trench, is 10.935 kilometres. Enceladus has an ocean nearly 3 times deeper than the deepest point of Earth's oceans, and it is currently venting a portion of that ocean into space at 400 meters per second. This is the Enceladus paradox, as I think of it. The smallest, coldest, most inconsequential seeming moon in the Saturn system turned out to be the most hydrologically active body in the solar system after Earth. But knowing the ocean existed was not the same as understanding what was in it. And what was in it? That question would take another decade to begin answering, and its answers, when they came, were nothing like anyone expected. Astrobiology, the science of looking for life beyond Earth, operates on a framework that is deceptively simple but extraordinarily demanding. Life, as we understand it, requires a small number of ingredients, every single one of which must be present simultaneously and persistently. Remove any one of them and the chemistry of biology becomes impossible. The checklist has 5 items: liquid water, a source of energy, the right chemistry, stability over geological time, and the right temperature range. It sounds simple. It eliminates almost every object in the solar system immediately. Liquid water, confirmed on Enceladus by 2006. Energy, provisionally confirmed by the hydrothermal signatures, but not yet quantified. The right chemistry, unknown. And this was the critical unknown: stability. Completely unknown. A liquid ocean is useless for the origin of life if it only lasts 1 million years. Life on Earth took approximately 400 million years to appear after the planet formed. Chemistry needs time, temperature range, implied by the liquid water itself, but the ocean could theoretically be barely above freezing or anomalously hot, which mattered enormously. The chemical story of Enceladus is one of the most methodical scientific investigations of the 21st century. It was not a single dramatic discovery. It was a series of findings, each one closing a gap in the checklist, each one raising the probability that something biological was possible in that hidden ocean. And each one also raised a harder question, not whether the ingredients existed, but whether they were present in quantities sufficient to sustain life, and whether they had been present long enough. The first major chemical revelation came in 2017 when a team analysing Cassini's mass spectrometer data found methane in the plumes in concentrations far too high to be explained by simple chemistry. Methane on its own is not unusual. It is one of the most common molecules in the cosmos. But the methane in Enceladus's plumes was accompanied by carbon dioxide and molecular hydrogen in proportions that, when modelled together, strongly resemble the output of a specific biological process, methanogenesis. On Earth, methanogenic microbes are ancient, primitive, and deeply embedded in the story of the biosphere. They are found at hydrothermal vents on the ocean floor, in the guts of cattle, and in the anaerobic sediments of the deep sea. They produce methane by combining hydrogen with carbon dioxide, and the ratio of these gases in Enceladus's plumes matched the thermodynamic fingerprint of methanogenesis with uncomfortable precision. Uncomfortable because it did not prove biology. It proved that the conditions for this particular form of metabolism existed. There could be an abiotic explanation, non-biological chemistry, that produces the same gases in the same proportions. The hydrogen, scientists noted, could also arise from serpentinization, a geological process in which water reacts with iron-bearing minerals at the seafloor and produces hydrogen as a byproduct. This reaction also generates heat. It was exactly the kind of process expected at a hydrothermal vent environment. And here the story deepened. In 2015, Cassini had already found silica nanoparticles in Saturn's E-ring, particles 2 to 8 nanometers in diameter, with a very specific chemical composition that pointed to one and only one formation pathway: high-temperature alkaline water between 90 and 110 degrees Celsius, dissolving silicon-rich rock, and then cooling rapidly to precipitate the silica. This is not a process that happens in cold water. This is a process that requires a hydrothermal vent system where superheated water circulates through rock, picking up minerals, and then erupts into a cooler environment. On Earth, this process occurs at places like the Lost City hydrothermal field in the middle of the Atlantic Ocean, a system of carbonate towers rising 60 meters from the seabed, bathed in warm alkaline water and teeming with microbial life. Enceladus, it appeared, had something like the Lost City at its ocean floor, a system of vents where the rocky core, warmed by tidal forces from Saturn, heated the ocean water above it to temperatures that could approach 100 degrees Celsius and sent mineral-rich fluid convecting upward through the water column and ultimately through the ice cracks and into space. This was the energy problem solved. Not a tentative maybe, a geochemically confirmed hydrothermal system, pumping heat into the base of a global saltwater ocean. 3 boxes on the checklist. Liquid water confirmed. Energy confirmed. But the chemistry box, the one that required not water and heat, but the specific organic molecules that life is built from, that one remained stubbornly incomplete. scientists knew organic molecules were present in general terms. Cassini had detected organic compounds in the plumes, but the resolution of those measurements was limited. The specific molecules remained elusive, and then, in 2023, that changed. Two separate studies, published within months of each other, reported findings that, when placed side by side, created something close to a biological shopping list, and confirmed that Enceladus was stocking every shelf on it. The first study, led by Frank Postberg at Freie Universität Berlin and published in Nature in June 2023, reported the detection of phosphorus in Enceladus's ocean. The finding arrived via Cassini's cosmic dust analyzer data, which had captured icy particles from Saturn's E-ring, particles that originated in Enceladus's plumes and had been dispersed into orbit. When those particles were analysed, they revealed sodium phosphates. Sodium, oxygen, hydrogen and phosphorus, chemically bonded in a configuration that could only have formed in liquid water, phosphorus. The molecule that forms the backbone of DNA and RNA. The molecule that powers ATP, adenosine triphosphate, the energy currency of all known life on Earth. The molecule so essential to biology that its absence has historically been considered a definitive indicator that an environment cannot sustain life. It had never before been detected in an ocean beyond Earth. The concentration was not a trace. Laboratory experiments and geochemical modelling, conducted by the international team as part of the same study, suggested that Enceladus Ocean contains phosphate at concentrations at least 100 times higher than Earth's oceans. 100 times higher. The researchers modelled why this was the case, and the explanation was elegant. Enceladus's carbonate-rich ocean chemistry, combined with the specific mineralogy of its rocky core, creates conditions in which phosphorus leaches from rock into water with exceptional efficiency. The same conditions, the team noted, might exist on other icy ocean worlds throughout the solar system. The second study, published in December 2023 in Nature Astronomy, and led by Jonah Peter at Harvard University, in collaboration with JPL's Kevin Hand, reported the detection of hydrogen cyanide in Enceladus's plumes. I want to be direct about what this means, because the molecule's name may trigger an instinctive recoil. Hydrogen cyanide is toxic. It is the compound that makes apple seeds mildly poisonous, and that was used as a chemical weapon in the 20th century. But in the chemistry of life's origins, it is not a poison, it is a seed. It is, as Peter described it in the study, the starting point for most theories on the origin of life, and the Swiss Army knife of amino acid precursors. Its molecules can be assembled in dozens of different configurations to produce the amino acids that proteins are built from. It is the most chemically versatile of the pre-biological building blocks, and it requires specific conditions to form, conditions that point to active chemistry, not passive geological equilibrium. Finding hydrogen cyanide in Enceladus's plume was not evidence of life, it was evidence of the chemistry from which life begins. The distinction matters enormously, and we will return to it. But alongside the phosphorus, alongside the hydrothermal vents, alongside the salt water and the silica nanoparticles and the methane, it was one more row of evidence filling in that checklist. 3 of 5 boxes were now deeply confirmed. Chemistry had joined water and energy. The remaining 2, stability over geological time, and whether the ocean had existed long enough, hung open like a wound. On the 1st of October 2025, a paper appeared in Nature Astronomy that I find, and I want to be precise about this word, transformative. Not because it contained a single finding of exceptional weight, but because of what its findings represented collectively. The arrival of a new level of chemical complexity in the Enceladus story, one that shifted the conversation from could life exist here to something significantly harder to dismiss. The lead author was Nozer Kawaja of Freie Universität Berlin, who heads a research group focused on the chemistry of outer solar system environments. The broader team included Fabian Klenner, a postdoctoral researcher at the University of Washington, who had spent years developing new analytical methods for interpreting Cassini's cosmic dust analyzer data, methods more sensitive than those available when Cassini was actively operating. The paper was entitled, simply, Detection of Organic Compounds in Freshly Ejected Ice Grains from Enceladus. The source of data was a specific and highly valuable subset of the Cassini archive, measurements taken during a close flyby of Enceladus's plumes, not from the dispersed E-ring material, but from ice grains captured within minutes of their ejection from the Moon's interior. These are grains that have been exposed to the open vacuum of space for the shortest possible time. Material that is, in the language of the paper, fresh. Material that most closely reflects the composition of the ocean itself, before ultraviolet radiation or chemical reactions in space begin to alter it. What Klenner and his colleagues found in those fresh grains was a vocabulary of organic chemistry that had not been detected before. Esters, alkenes, ether compounds. The names may not immediately illuminate what this means, so let me translate them into structural terms. Esters are organic compounds formed when an acid reacts with an alcohol. They occur throughout nature. They are responsible for the fruity aromas of overripe fruit, but more importantly, many ester bonds are central to the chemistry of living systems. Ethers are a related class of compound in which an oxygen atom links two carbon chains together, and this is the key sentence from the paper. Esters and ethers can be part of lipids, and lipids, the fats and oils and phospholipid bilayers of cell membranes, are foundational to all life on Earth. A cell membrane is, at its most basic structural level, a lipid bilayer, two sheets of lipid molecules arranged tail to tail, creating a barrier that separates the interior chemistry of the cell from the external environment. Without lipid membranes, there are no cells. Without cells, there is no life as we know it. Enceladus's ocean contains the precursors of cell membranes. I want to sit with that for a moment because I think the pace of scientific discovery sometimes allows findings of extraordinary weight to pass without the pause they deserve. We have found, in the active plumes of a moon smaller than the United Kingdom, not merely water and warmth, but the molecular architecture from which cellular life could in principle assemble itself. We have found the lipid precursors, we have found the phosphorus for the DNA backbone, we have found hydrogen cyanide as the amino acid precursor, we have found hydrogen for the metabolic energy source, we have found hydrothermal vents to drive the chemistry. It appears that Enceladus has all the ingredients for life as we know it, said Klenner in the press release accompanying the paper. This is a sentence from a scientist who spent years stress testing that conclusion and found it held up. The detection was not a surprise to Kawaja. His research group had been preparing the analytical approach for this kind of measurement for years. What surprised him was the richness of the catalogue. We found a rich organic inventory in Enceladus's plume, he said. The word rich is doing significant scientific work in that sentence. It does not mean traces. It does not mean ambiguous signals at the edge of instrumental sensitivity. It means a diverse and abundant array of chemically distinct molecules present in quantities sufficient to be unambiguously characterized. The 2023 hydrogen cyanide study had found something equally important in the energy domain. JPL's Kevin Hand described the finding in a metaphor that stuck with me. If methanogenesis, the methane-producing process identified in the 2017 analysis, is like a small watch battery in terms of energy, then the oxidized organic compounds identified in the 2023 analysis suggest the ocean of Enceladus offers something more akin to a car battery, a large, powerful, sustained energy source capable of supporting a diverse range of metabolic pathways, not just the primitive methanogenesis of the early Earth, but a broader spectrum of possible biochemistries. This matters because the origin of life is not merely a chemistry problem. It is an energy problem. Life does not spontaneously emerge from the right chemical ingredients sitting in a pool. It requires a sustained thermodynamic gradient, a difference in energy states that allows chemical reactions to run continuously in a particular direction, building complexity instead of dismantling it. Hydrothermal vents provide exactly this: hot water meeting cool water, alkaline fluid mixing with acidic seawater, mineral surfaces providing catalytic scaffolding. On Earth, the leading theories of the origin of life most notably those associated with the Lost City hydrothermal system, and the work of Nick Lane and Michael Russell placed the first living chemistry at precisely these kinds of interfaces. Enceladus has that interface in its ocean right now, 1.272 billion kilometres from Earth. The 2025 study from the University of Oxford and the Southwest Research Institute closed the last major structural gap in the habitability argument. Until November 2025, the question of whether Enceladus's ocean was stable, genuinely stable, in balance, sustainable over the millions of years required for life to arise, had remained open. The south pole of Enceladus, with its active tiger stripes and erupting plumes, was clearly losing heat. But how much heat? And was it balanced by the tidal input from Saturn? Without that balance, the ocean could be ephemeral, a geological accident in its middle phase, burning brightly now but destined to freeze. The Oxford-led study, published in Science Advances in November 2025 and led by Dr. Georgina Miles of the Southwest Research Institute and Oxford's Dr. Carly Howitt, addressed this question with a method nobody had attempted before. The team compared Cassini infrared observations of Enceladus's North Pole, taken a decade apart in 2005 and 2015, to measure the temperature of the surface at the North Pole. The North Pole of Enceladus was considered geologically inactive. No plumes, no tiger stripes, no obvious heat source. But when the team modelled what the surface temperature should be at the North Pole, based purely on solar input and radiation to space, they found the observed temperature was approximately 7 degrees Kelvin warmer than predicted. 7 Kelvin is not a large number in absolute terms, but in the context of Enceladus, it is enormous. That 7-degree excess could only be explained by heat leaking from the interior, conductive heat flow from the subsurface ocean radiating through the ice shell and warming the surface at the North Pole. The measured heat flow rate was 46 milliwatts per square metre. That sounds trivial, but across the entire North Polar region, it adds up to a significant contribution to the global heat budget. Combined with the previously measured heat loss from the active South Pole, the total heat escaping from Enceladus's surface is approximately 54 gigawatts. 54 gigawatts. For context, that is roughly the combined power output of 10,500 modern wind turbines, or the electrical generating capacity of a small nation escaping silently through the ice of a moon that for centuries was classified as an unremarkable frozen rock. And here is the crucial number. Theoretical models of tidal heating in Enceladus, based on Saturn's gravity and Enceladus's orbital characteristics, predict a heat input of 50 to 55 gigawatts. The observed heat loss of 54 gigawatts falls squarely within that range. The Moon is in thermal equilibrium. It produces as much heat as it loses. The ocean is stable. Not temporarily, not coincidentally, structurally, persistently, sustainably stable, maintained in its liquid state by a balance of forces that could, in principle, persist for hundreds of millions of years or more. Understanding how much heat Enceladus is losing on a global level is crucial to knowing whether it can support life, said Dr. Howitt. This new result supports Enceladus's long-term sustainability, a crucial component for life to develop. All 5 boxes on the checklist were now confirmed, or confirmed to within the observational limits of the data available. Liquid water, a global ocean 26 to 31 kilometers deep. Energy, hydrothermal vents on the ocean floor confirmed by silica nanoparticles. Chemistry, phosphorus, hydrogen cyanide, esters, ethers, alkenes, methane. Hydrogen, a complete biological pharmacy. Temperature, the vents drive water to near 100 degrees Celsius at the floor. The ocean mean temperature is suitable. Stability, 54 gigawatts in, 54 gigawatts out. The ocean has been there long enough. The ocean will be there long enough. But here is the question that none of these findings, however extraordinary, can answer. Is something alive in that ocean right now? And if it is, how do we know? There is a particular quality to the dedication of a scientist who spends years analysing data from a mission that no longer exists. The Cassini-Huygens spacecraft launched from Cape Canaveral on the 15th of October 1997. It was, at the time, the largest and most expensive interplanetary probe ever built, a joint effort between NASA, the European Space Agency, and the Italian Space Agency, constructed at a cost of approximately $26 billion, weighing 5,712 kilograms at launch, and carrying 12 distinct scientific instruments designed to study Saturn, its rings and its moons in unprecedented detail. It took 7 years to reach Saturn, using a sequence of gravitational assists at Venus, Earth, and Jupiter to build the velocity required for the journey. It arrived in Saturn orbit on the 1st of July 2004. For 13 years, Cassini orbited Saturn. It circled the planet 294 times. It executed dozens of targeted flybys of Saturn's moons. It released the Huygens probe, which descended through the clouds of Titan in January 2005, and returned the first images from the surface of an outer solar system moon. It captured images that redefined our visual vocabulary for the outer solar system. Images of Saturn's rings in backlit silhouette, of moonrise above Titan's hydrocarbon seas, of Enceladus with its south pole ablaze in the infrared. And then, on the 15th of September 2017, following a final, deliberate sequence of maneuvers designed to prevent the spacecraft from accidentally contaminating the moons it had studied, Cassini entered Saturn's atmosphere and was destroyed. The mission was over. But the data was not. Frank Postberg grew up in Germany and trained as a physicist. By the time Cassini arrived at Saturn, he was already working on the Cosmic Dust Analyzer, the CDA, the instrument that would become the primary tool for the most consequential chemical discoveries in the mission's post-operational life. The CDA worked by capturing individual dust particles and ionizing them on impact, allowing a mass spectrometer to read the composition of each particle in real time. It was sensitive to a remarkable degree, capable of identifying the molecular weight of compounds within a captured grain at masses smaller than a few hundred daltons. But its sensitivity was not unlimited, and its data, during the years of active operations, was interpreted using the analytical tools and chemical libraries available at the time. The problem, and this is where the story becomes a story about patience, is that the analytical tools improved dramatically after the mission ended. Postberg, by 2022, was leading a research group at Freie Universität Berlin, with a team that included Fabian Klenner, a postdoctoral researcher who had developed new computational methods for interpreting Cassini CDA spectra. These methods involved more sophisticated pattern matching algorithms and a more comprehensive chemical reference library. Essentially, a more complete dictionary against which to compare the signals captured by the instrument. The data had been sitting in NASA's planetary data system for years, available to any scientist in the world. The same measurements, the same archive, but read through a more powerful lens. Much of the CDA data still isn't analysed, Klenner said in October 2025, shortly after the publication of the Fresh Grain Organics paper, and I'm so excited about what it may reveal next. The discovery of phosphorus is the one that will stay with me, not because it was the most dramatic compound, but because of what it required, because of the specific combination of tenacity, creativity, and methodological innovation that led to a finding that changed the entire shape of the habitability debate. Phosphorus had been theoretically predicted to exist in Enceladus's ocean. The geochemical models were clear. Given the mineral composition of the rocky core and the alkaline character of the ocean, phosphate should be present. Christopher Glein of the Southwest Research Institute had published modelling papers indicating this in 2022. But modelling is not detection, and detection requires signal. Phosphorus, the least abundant of the essential bioelements, was expected to appear in low concentrations, producing weak signatures that could easily be drowned out by the stronger signals of sodium, chlorine, and the dominant organic compounds. The breakthrough came through a methodological choice. Instead of analysing data from Cassini's direct plume flybys, where the particle sample was rich but the flight time was short and the statistics were limited, Postberg's team chose to focus on a different dataset, the E-ring crossings. Every time Cassini crossed Saturn's E-ring, which was frequently, the CDA sampled thousands of tiny ice particles that had originated in Enceladus's plumes and had since dispersed into orbit around Saturn. These particles were older. They had been in space for days or weeks, but there were vastly more of them, and in that large statistical sample, the signature of sodium phosphate emerged, not as a single spectacular detection, as a persistent, reproducible pattern across thousands of individual particle measurements. Postberg described the finding with restraint, but the restraint barely concealed the weight of what he was saying. Now, this new result reveals the clear chemical signature of substantial amounts of phosphorus salts inside icy particles ejected into space by the small moon's plume. It's the first time this essential element has been discovered in an ocean beyond Earth. The first time, an ocean beyond Earth, containing the element without which DNA cannot exist. I want to spend a moment with the person at the centre of this story, because science is done by people, and the human texture of it matters. Fabian Klenner had been working on Cassini CDA data for years before the 2025 organics paper. His doctoral work, his postdoctoral research, his entire early career had been built around a spacecraft that no longer existed, around data that had been collected, in some cases, years before he was a professional scientist. The plume flyby data that yielded the esters and ethers and alkenes, the data that appeared in the October 2025 Nature Astronomy paper, was collected in some cases during flyby campaigns conducted in 2008 and 2011. The data was 17 years old when its most significant secrets were extracted. There is something quietly extraordinary about this fact. The Cassini mission ended in fire in 2017. The spacecraft burned, the instruments ceased to transmit, the teams dispersed to other projects, other missions, other institutions, and yet the data persisted, archived, digitized, publicly available, waiting. And scientists like Klenner, working at universities in Berlin and Seattle and Stuttgart, returned to it year after year, applying new tools to old signals, patient in the way that the best science requires patience. It's phenomenal to continue learning from the Cassini mission, Klenner said. Having clear evidence of a variety of organic compounds from inside an extraterrestrial water world is incredible. He will start a position as an assistant professor at the University of California, Riverside. He is, by any reasonable assessment, at the beginning of his career, and the most consequential discovery of his career so far was made by listening more carefully to data that everyone else had already heard. Linda Spilker, who served as the Cassini project scientist at JPL, and who dedicated 30 years of her professional life to the mission, put it this way when the phosphorus discovery was announced. This latest discovery of phosphorus in Enceladus's subsurface ocean has set the stage for what the habitability potential might be for the other icy ocean worlds throughout the solar system. Now that we know so many of the ingredients for life are out there, the question becomes, is there life beyond Earth, perhaps in our own solar system? I feel that Cassini's enduring legacy will inspire future missions that might eventually answer that very question. She did not say there is life. She said the question is alive. And that is, in the discipline of science, the most honest and the most thrilling thing you can say. But there is a complication buried inside all of this. One that the enthusiastic headlines from 2023 and 2025 sometimes overlooked. The ingredients are present, the conditions are right, the checklist is complete. And yet, and this must be said clearly, not one of these discoveries, individually or collectively, constitutes evidence of life. They constitute evidence of habitability. These are profoundly different things. The ocean of Enceladus may be the most habitable extraterrestrial environment ever characterized, and it may be completely sterile. Why? Because habitability and inhabited are not the same. Earth has been habitable for approximately 4.5 billion years. For roughly the first 400 to 600 million of those years before life appeared, it was habitable and uninhabited. Chemistry requires time to self-organize. The origin of life, the transition from non-living chemistry to self-replicating, metabolizing, dividing biological entities is not a guaranteed outcome of having the right ingredients. We do not know how difficult it is. We have exactly one data point, us. And from one data point you cannot calculate a probability. This is the origin problem. And it is the deepest question that Enceladus, for all its extraordinary chemistry, cannot yet answer. At some point in the history of the Enceladus investigation, the question stopped being a question only about a small moon in the outer solar system. It became a question about the nature of life itself, about whether biology is a universal phenomenon or a local accident, about whether the kind of chemistry that assembled itself into a living cell here, on this particular rock, 3.8 billion years ago, is the kind of chemistry that the universe routinely generates, or whether it was a fluke of cosmic proportions that will never be repeated. This is the question that the scientific community calls the Fermi question, or in its most precise formulation, the Drake equation problem, the question of whether we are alone. And it has, since the 1960s, been answered with a frustrating silence. We have found no signals from extraterrestrial civilizations. We have found no fossils on Mars. We have found no unambiguous biosignature on Venus, or Titan, or Europa, or anywhere else in the solar system we have visited. The silence is not proof of absence. The universe is incomprehensibly vast, and we have searched only a negligible fraction of it. But the silence is also not nothing. It accumulates, and it weighs. If life exists in the ocean of Enceladus, if some form of microbial chemistry has taken root in the warm, mineral-rich, chemically complex water beneath that ice, then the silence breaks. Not spectacularly, not with a signal from a distant civilization or the image of an alien landscape. Silently, chemically. A few molecules in a plume, but the implication would be seismic. Here is why. If life arose independently on Enceladus, if it did not come from Earth via a meteorite impact that ejected biological material into space, which is theoretically possible but extraordinarily improbable, then it arose from chemistry in a warm ocean, which is essentially the same chemistry and the same environment in which life arose on Earth. Two independent origins of life, two data points instead of one. And from two data points, you can begin to calculate a probability. You can begin to say that life, under the right conditions, is not a fluke. It is an outcome, perhaps an inevitable one. Carl Sagan expressed this idea 40 years ago in a formulation that has aged better than almost any other speculation in modern science. The origin of life on suitable planets seems built into the chemistry of the universe. He said it speculatively, from intuition and analogy, because there was no evidence for it. Evidence of life on Enceladus would transform it from a beautiful speculation into an empirical conclusion. The implications cascade outward in directions that are frankly difficult to absorb all at once. If life arises wherever the conditions are right, then consider the conditions that have been confirmed to be right. Enceladus, Europa, Jupiter's moon, which also has a subsurface ocean, confirmed by the Galileo spacecraft and now under active investigation by NASA's Europa Clipper mission, launched in October 2024 and currently in transit to Jupiter. Titan, Saturn's largest moon with its hydrocarbon lakes on the surface and a possible water ammonia ocean in its interior. Ganymede, Callisto, potentially Ceres, potentially dwarf planets in the Kuiper Belt. And that is only our solar system. There are an estimated 200 billion solar systems in the Milky Way galaxy alone. Many of those systems have icy moons. Many of those icy moons will have liquid water oceans heated by tidal forces from their parent planets. Many of those oceans will have the organic chemistry and the geothermal energy to sustain life. The number of potentially habitable environments in the universe, if Enceladus is anything to go by, may be in the hundreds of billions, perhaps the trillions. Life under this framework is not the exception, it is the rule. But there is a harder and more uncomfortable implication that rarely gets raised in the popular coverage of these discoveries. If life is common, if it arises readily in subsurface oceans across the galaxy, then the Fermi paradox deepens dramatically. The silence in the sky in that scenario is not because life is rare. It is because life almost never produces technological civilizations. or because technological civilizations are short-lived, or because the universe is so vast that even 1 billion technological civilizations could pass their entire existence without ever encountering one another. The discovery of microbial life on Enceladus would not resolve the question of whether we are alone in any meaningful sense. It would simply restate it at a new level of depth. We are alone here, in our solar system perhaps, but the silence in the sky, if life is on Enceladus, becomes an even louder question than it was before. And there is a third implication that is, to my mind, the most consequential of all, and the one that planetary protection scientists spend the most time worrying about. If life exists on Enceladus, life that arose independently, from a separate origin, with potentially a separate biochemistry, separate amino acid chirality, separate genetic encoding mechanisms, then it represents a second example of how life can work. A second solution to the problem of self-replication and metabolism. The comparison of that solution with ours would tell us, within the span of a generation of scientific study, more about the deep logic of biology than the entire history of terrestrial life science has been able to tell us in 3 centuries, the reason biology works the way it does, why DNA uses 4 bases, why proteins fold the way they fold, why the cell membrane operates by lipid bilayers, why metabolism runs on ATP, is not fully understood. We know how life works on Earth, we do not know why. We cannot distinguish between aspects of terrestrial biology that are universal laws of chemistry and aspects that are historical accidents of our specific evolutionary pathway. A second biosphere would give us that ability. It would separate the contingent from the necessary. It would tell us what life, everywhere in the universe, has in common. This is why Christopher Glein of the Southwest Research Institute called the phosphorus discovery stunning for astrobiology rather than merely important. This is why Klenner said incredible rather than notable. This is why, when I read the October 2025 paper from his team, I found myself doing what I almost never do when reading scientific literature, stopping, putting the paper down, and sitting with it for a few minutes in silence. Not because the paper said life was there, it didn't, but because of how close the evidence had come to the edge of that conclusion, and because of what it would mean if it crossed it. The stakes here are not merely scientific. They are philosophical, religious, and existential in a way that almost no other scientific question can match. The question of whether we are alone in the universe has animated human thought since the first moment humans looked up at the sky and wondered what those lights were. Every cosmology, every theology, every philosophy of human uniqueness has an implicit answer to that question embedded in it. If Enceladus has life, every one of those answers requires revision. And here is what makes the situation so extraordinary, so frustrating, so perfectly and characteristically scientific. We can see the evidence accumulating. We can watch the checklist being completed. We can calculate the probabilities. We can read the chemistry. We cannot, with any mission currently operational or approved, actually look inside the ocean and tell whether anything is swimming in it. That gap between what we know and what we need to know is the gap that the next chapter of this story must try to cross. Science has a habit of being inconveniently honest. The more you study a subject, the more clearly you understand not only what has been established, but what remains unresolved. And sometimes the unresolved questions are more significant than the answers. The case for life on Enceladus is the strongest extraterrestrial habitability argument in the history of planetary science. It is not, however, a closed case, and the complications in the story are not footnotes. They are load-bearing walls of the scientific argument, and they deserve to be addressed with the same rigour as the headline discoveries. The first complication is what planetary scientists call the abiotic mimicry problem. Every molecule detected in Enceladus's plumes can in principle be explained by non-biological chemistry. Methane, produced by serpentinization and other geological processes. Hydrogen, the byproduct of water reacting with iron-bearing minerals at depth. Hydrogen cyanide, formed when nitrogen and carbon-bearing compounds react in high-energy environments. esters, ethers, alkenes, organic synthesis in hydrothermal systems is extraordinarily prolific on its own, without any biological catalysis. The amino acid precursors, the lipid precursors, phosphorus compounds, all of them have abiotic formation pathways that are chemically plausible. This is not a defect in the science. This is the correct situation for this stage of the investigation. The molecules we have detected are necessary for life, but they are not sufficient proof of it. The presence of a kitchen, fully stocked, tells you nothing about whether anyone has been cooking in it. JPL's Kevin Hand, who led the team that found hydrogen cyanide and the oxidized organic energy sources in 2023, was scrupulous about this in his public communication. Scientists are still a long way from answering whether life could originate on Enceladus, he said. The word originate is important here, not whether life could persist, but whether it could have started. These are two different questions. Persistence is in some ways the easier problem. Given an existing metabolism, given an existing self-replicating molecule, maintaining it in a warm, energy-rich environment is plausible. But the jump from complex chemistry to self-replication, the formation of the first RNA-like molecule, the first enclosed cell membrane, the first catalytic cycle, remains the deepest unsolved problem in science. We do not know how difficult it is, because we have never observed it happening. The second complication is temporal uncertainty. The Oxford study from November 2025 confirmed that Enceladus's ocean is currently in thermal equilibrium. It is losing as much heat as it gains. But currently, in a planetary science context, means the present epoch, not the entire history of the Moon. The question of how long the ocean has been liquid remains, as the Oxford authors explicitly stated, still uncertain. Life on Earth took several 100 million years to appear. If Enceladus's ocean is only a few 100 million years old, life might not have had time to begin. If it is 1 billion years old or more, some models suggest it could be, then the time constraint is removed. This is not a trivial distinction. The age of the ocean is a critical parameter in the habitability argument, and it is one we cannot currently measure with available data. The best estimate involves modelling the internal heat budget over geological time, which depends on the orbital evolution of Enceladus. Specifically, how its orbital resonance with the other moon Dione has changed over time. This is an ongoing area of research, and different models produce different ages. The range currently spans from perhaps a few 100 million years to something approaching the age of the solar system itself, roughly 4-5 billion years. The third complication is what I would call the access problem, and it may be the most practically significant of all. Enceladus is generous with its ocean in one specific and extraordinary way. It actively ejects ocean water into space, where a spacecraft can fly through it and taste it. No other ocean world in the solar system offers this. Europa has a fractured surface and possible plumes, but nothing remotely as well characterized or persistent as the Enceladus tiger stripe plumes. Titan's hydrocarbon seas are surface features, not subsurface oceans. The Enceladus plumes represent a direct sample of the ocean interior, with only a brief transit through a few kilometers of ice and the plume environment before reaching space. But brief transit is not direct access. The ice grains that Cassini sampled had been through the plume venting process, accelerated to 400 meters per second, and flash frozen. They had spent time, minutes to hours, in the vacuum of space before being captured. The organic compounds in them are real, but they are a filtered, fractionated, partially processed representation of the ocean chemistry. Delicate biological molecules, proteins, nucleic acid fragments, intact cell membrane material, might not survive the energetics of the plume process and the exposure to ultraviolet radiation. A biosignature in the plume might be destroyed before we can detect it. John Peter put it this way: the oxidized compounds found in the 2023 study suggest there are many chemical pathways to potentially sustain life, which is correct, but note the word potentially. Even the most sophisticated mass spectrometry available on Cassini, a spacecraft designed in the 1990s and launched in 1997, had sensitivity limits that placed certain classes of biosignature beyond its detection threshold, specifically intact cells, intact proteins, intact nucleic acid sequences. The instruments aboard Cassini were not designed to look for life. They were designed to characterize chemistry. The distinction matters enormously. There is also a contamination argument, which receives surprisingly little popular attention, but which dominates planetary protection discussions at NASA and ESA. When we send a spacecraft to sample the environment of a potentially inhabited world, we must be certain that any biology we detect came from that world and not from Earth. Cassini was not sterilized to the standards required for a mission with a meaningful probability of biological contact with a habitable environment. It was not expected to encounter an active ocean world. Its microbiological contamination levels, while managed according to the planetary protection protocols of the 1990s, would not meet the standards that would be required for a dedicated life detection mission in the 2020s or 2030s. This does not mean Cassini contaminated Enceladus. It was deliberately destroyed in Saturn's atmosphere, precisely to avoid any risk of crashing into a habitable moon. But it does mean that the detection claims must be carefully bounded. We can say with high confidence what molecules are present in the plume. We cannot yet say whether any of those molecules are products of biology or geology. The fourth complication is philosophical rather than technical, but it is, in a sense, the most important. We do not have a definition of life that is universally accepted. On Earth, we recognize life by a cluster of characteristics: self-replication, metabolism, response to stimuli, growth, adaptation. But these characteristics are derived entirely from studying life as it exists here. Life on Enceladus, if it exists, may have arisen from a different chemical origin, different amino acid chirality, the handedness of molecules, different coding mechanisms, different metabolic pathways. It might not replicate in the way we expect. It might not have the same chemical signature we associate with terrestrial biology. It might be using chemistry we have never encountered and would not recognize as biological even if we measured it directly. This is the most profound problem, not the detection problem, not the sterilization problem, not the temporal problem. The possibility that life, in its second known incarnation, might not look like life from where we stand. None of these complications reduce the significance of what has been found between 2023 and 2025. They contextualize it. They place the headlines in their proper scientific frame. The case for Enceladus as the most compelling extraterrestrial habitable environment ever characterized remains, after every complication is applied, the strongest case in the history of astrobiology. The checklist is complete. The chemistry is extraordinary. The evidence, assembled over decades of Cassini data analysis, is as robust as current technology permits. What remains is not a question of whether the ingredients are there. The ingredients are there. The question is whether the recipe is being cooked. And to answer that, we need to go back. Not with a flyby spacecraft, not with archived data. We need to go back with instruments designed from the ground up to look for life. There is a spacecraft being designed right now that, if it launches on schedule and arrives on schedule and functions as designed, could answer the oldest and most consequential question in human intellectual history. Are we alone? It is called the Enceladus Orbelander. The concept was proposed during NASA's Planetary Science Decadal Survey of 2023-2032, the once-per-decade process by which the planetary science community assesses priorities, allocates resources, and charts the direction of major missions for the coming decade. The 2023 Decadal Survey, titled Origins, Worlds and Life, ranked the Enceladus Orbelander as the second highest priority flagship mission of the decade, behind only a Mars sample return. A flagship mission is NASA's highest budget category of space exploration, not a small CubeSat or a moderately priced Discovery mission, but a major multi-billion-dollar endeavour of the scale of Cassini, Voyager, or the James Webb Space Telescope. The Orbelander concept involves two phases. In the first phase, lasting approximately 200 days, the spacecraft would orbit Enceladus, flying repeatedly through the plumes and collecting data with an instrument suite designed specifically for the detection of life and the characterization of ocean chemistry at sensitivities orders of magnitude beyond what Cassini could achieve. This orbital phase would build on everything Cassini found, but with instruments whose capabilities reflect 3 decades of advancement in mass spectrometry, biosignature detection, and astrobiology-focused analytical chemistry. In the second phase, the spacecraft would descend to the surface of Enceladus, specifically to the south polar terrain near the Tiger Stripes, and land. As a surface lander, it would sample the freshly deposited snow that continuously falls back onto the surface around the active vents. This snow is ocean water. It is the most accessible sample of a subsurface ocean anywhere in the solar system, deposited at the surface and available for collection without drilling, without penetrating an ice shell, without any of the technological challenges that a Europa or Ganymede lander would face. The lander would analyse these snow samples with instruments capable of detecting intact organic molecules at concentrations as low as parts per trillion, sensitive enough, in principle, to detect the chemical signatures of individual cells. The instrument suite proposed for the Orbolander includes a high-resolution mass spectrometer capable of identifying molecular masses to within a fraction of a Dalton, allowing it to distinguish between biological and abiotic formation pathways for specific compounds. It includes a microscope capable of imaging individual particles at micrometer resolution, sufficient to identify cellular morphologies. It includes a fluorescence detector sensitive to DNA-binding dyes, which would glow in the presence of nucleic acids. It includes an amino acid analyzer capable of determining the chirality of amino acids, whether they are left-handed or right-handed, because Earth life uses almost exclusively left-handed amino acids, and a random chemical environment would produce equal mixtures. The preponderance of one hand over the other in Enceladus's snow would be one of the strongest possible indicators of biology. The European Space Agency has also designated Enceladus as a top priority. In ESA's Voyage 2050 long-term science plan, an Enceladus mission was selected as a candidate for the agency's first large-class mission of the program, which would follow the JUICE mission to Jupiter, the LISA Gravitational Wave Observatory, and the New Athena X-ray telescope. ESA's conceptual approach differs from NASA's, but converges on the same imperative: a dedicated life detection-focused spacecraft with state-of-the-art instruments, sent specifically to the place in the solar system where the evidence for habitable conditions is strongest. The timeline is not near. A flagship mission of this scope takes a decade or more from approval to launch. Given the current budgetary landscape at NASA and the competing priorities of Artemis, Europa Clipper, and Mars sample return, the earliest realistic launch window for an Orbolander-type mission is the late 2030s to early 2040s. Arrival at Enceladus, given the transit time and the required gravitational assist trajectory, would be in the late 2040s to 2050s. The answer to the most important question in science may be 5 decades away from today, but in some ways that is an astonishingly short time. The question of life beyond Earth has animated human thought for at least 2,000 years, from Epicurus in ancient Greece to Bruno in the Renaissance to Lowell, and his Martian canals to the first detection of an exoplanet in 1992. For almost all of that time, the question was purely philosophical. We had no tools, no missions, no evidence to bear on it. Now, for the first time in human history, we have a specific location, a specific mechanism, and a specific technology roadmap that could, within the span of a single human lifetime, deliver a definitive answer. That is unprecedented, whatever the answer turns out to be. There is one more development that belongs in this chapter, and it concerns not Enceladus, but what Enceladus teaches us about the rest of the solar system and the universe beyond it. The discovery that Enceladus's ocean is phosphorus-rich led the team, in their 2023 paper, to perform geochemical modelling of other ocean worlds. The models suggested that the same carbonate water rock chemistry that produces high phosphate concentrations in Enceladus's ocean would also operate in any icy moon where liquid water has prolonged contact with carbonaceous rock and where the initial water ice contained carbon dioxide, a common composition in the outer solar system, Europa, Ganymede, Titan's interior, Pluto's possible ocean. The moons of Uranus currently being investigated as a target for NASA's Uranus Orbiter and Probe mission, the highest priority flagship of the current decadal survey. These worlds, the models suggest, may all contain phosphorus-rich oceans, if Enceladus is the template. If the conditions that make it habitable are not unique but representative of a class of icy ocean worlds that extends throughout the solar system, and, by extension, throughout the galaxy, then the implications are not merely exciting. They are foundational. They suggest that the outer solar system is not a frozen, sterile desert, but a distributed biosphere in waiting, a set of independent experiments in the chemistry of life, each one sealed beneath kilometers of ice, each one erupting or dormant, or somewhere in between, each one carrying its ocean forward through time until something, a spacecraft, a scientist, an instrument sensitive enough to tell the difference between a lipid and a geological deposit, finally comes close enough to ask the question directly. This is where the science stands, at the edge of the most consequential expedition in human history. Not quite ready to leave, not quite sure what it will find, certain at last that it is worth going. The ocean of Enceladus has been there for a very long time. In geological terms, it predates every human civilization, every human species, every ancestor that walked on two legs. It predates the first multicellular life on Earth. It has been warm and dark and chemically complex, sealed beneath its ice, for periods of time that make the entire history of life on our planet seem brief by comparison. And for almost all of that time, it has been completely alone. The first thing that ever turned toward it was light. Sunlight so faint at 1.272 billion kilometers that it registers as little more than a bright star, cold and distant, barely warming the surface it touches. Then in 2004, something else arrived. A spacecraft, 12 instruments, 17 years of orbital mechanics. And behind those instruments, at the end of a signal chain stretching across more than 1 billion kilometers of space, human beings, curious, patient, desperate in the way that only curious creatures can be desperate to know. The data it gathered is still yielding answers. It will keep yielding answers for years, perhaps decades. The scientists who analysed that data found something they had suspected but could not confirm until now, that this ocean is ready, chemically ready, thermally stable, energetically active, stocked with every molecular ingredient that life, as we know it, requires to begin. And they found it not with a single dramatic revelation, but with a long, careful, methodical accumulation of evidence. One molecule at a time, one flyby at a time, one carefully re-examined dataset at a time. The finding did not come with certainty. It never does at the frontier. What it came with was a weight of evidence so substantial and a checklist so comprehensively filled that the burden of proof has, for the first time in the history of astrobiology, shifted. The question is no longer, could there be life on Enceladus? The question is, why would there not be? That is not the same as finding life. I want to be clear about that because honesty matters more than narrative momentum. We have not found life. We have found everything that life needs. We have found the kitchen, and the ingredients, and the stove, and we have found it running. Whether the chef has been there is still unknown, but consider what it means that the question has reached this point. In the whole sweep of human science, from the first taxonomy of living things by Aristotle, to the discovery of DNA's structure by Watson, Crick, Franklin, and Wilkins, to the detection of gravitational waves by LIGO, there has never been a moment quite like this one. A moment where the evidence for life beyond Earth was strong enough to make its absence more surprising than its presence. A moment where the answer genuinely could go either way, and where the technology to find that answer is already being designed. We are the generation that built the case. We are the generation that found the phosphorus, and the hydrogen cyanide, and the lipid precursors, and the thermal equilibrium. and the hydrothermal vents. We are the generation that established, with instruments that nobody designed to find life and yet found everything necessary for it, that a moon smaller than the United Kingdom is one of the most promising addresses in the known universe. The answer belongs to whoever comes next. The scientists who will board the metaphorical equivalent of whatever mission eventually reaches Enceladus, who will point their instruments at that warm, dark, erupting ocean and read what the chemistry says. The answer may be silence. It may be noise that resolves slowly into a pattern. It may be a left-handed amino acid where a random mix was expected. It may be a cellular shape under a microscope, pointed at a flake of fresh snow from a plume that has been erupting unceasingly for hundreds of millions of years. Enceladus has been broadcasting the ingredients of its ocean into space at 400 meters per second for longer than our species has existed. We only recently learned how to listen.